So we're ready to start Chapter 6. Chapter 6 discusses microbial growth. Now when we think about growth of microbes, you got to kind of look at it a little differently than what you normally think of the word growth. When you think about us growing, what does that mean? Getting bigger, right? Microbes don't really get bigger, all right? They're kind of born or produced, whatever word you want to use, the size that they're pretty much going to be. When we think about microbial growth, we're thinking about increasing the amount of microbes that we have. It's actually quite impressive how fast some microbes can grow. So I think it's worthwhile. Let me show you guys something. We have on this computer a video that was taken under a microscope of some microbes growing so that you can actually kind of see how they grow. Where we're going to be going after this chapter is looking at how we kill them. So in this chapter, we're going to get a very good appreciation of how fast they can grow, and then we're finally going to talk about how we can kill them. So what I want you to kind of think about, if this thing will eventually come up, is you guys know those commercials for things like toilet cleaners and Clorox wipes and all that, where they show you what's supposed to be bacteria under a microscope. They show you the little circle, and they squirt it and kill the bacteria in the circle, and it says it kills 99.9%, and there's just like 1 or 2 of them left. And that seems like, hey, that's really awesome, right? I just killed all of those. Well, let me show you how fast that can go from that one microbe that is sitting there. All right, can y'all see that? Okay, so this is sped up 440 times normal speed. So what we're going to view in just a few seconds would be maybe an hour, 2 hours. So let's just say we just cleaned our toilet. We did really awesome, right? We only left 1 or 2. Here's 20 minutes later. 45 minutes later. Oh, one was left over there off the screen too. An hour later. It's kind of impressive when you see it sped up like that. In your toilet, you can't kill it for good. So that's just E. coli. Let me show you another one. I know it took me a second to get them up here, but I usually think it's worthwhile for you to see it. This is staph. Staph's actually a little slower growing than E. coli, but still grows pretty fast. Why is this one red and the other one was green? They just injected different dyes into them. These are just fancier microscopes that are viewing this. This is Staphylo, so it's a little cluster. I think this looks like popcorn popping. So again, this is probably about 40 minutes, 45 minutes after you started. They grow a little slower. The only reason they're not taking over the screen is because they just grow up on top of each other. The E. coli was just kind of sprouting off everywhere. Okay? So now next time you think you've cleaned your toilet or you've cleaned your floor or something like that, just kind of remember those videos. But before I get you too frantic about, oh my God, my house is never clean, it is good for you to get some of those germs in you. Okay? You may not believe it from the things I say in my classroom, but when my kid eats something off the floor, I don't freak out. Just let her do it. Even when she was a baby, if her pacifier fell, I just stuck it back in her mouth. Because those mothers that are frantic are actually doing harm to their children. There was a lady in town about 2012, about 8 or 10 years ago. She was a nurse at Forest General, and she borrowed some cleaners to clean with after her child was born, and her child developed a very rare case of pneumonia and died because she had everything too clean. So you do need some of those germs. I'm just trying to make a point. All right. So we just watched a video of them growing. Let's think about how they grow. Now there's a couple terms we need to know. We're going to see, and you've probably heard me using some of this in MicroLab. Okay? And the first important term is a colony. And if we've used any plates at all, I can't remember if we have, you've probably heard me use that term. A colony is a little group of bacteria growing. So when you see a Petri dish and you see one little cluster of bacteria growing on there, that's one colony. Everybody in one colony belongs to one family. They're all genetically identical. That's the difference between eukaryotes and prokaryotes. They divide by binary fission, which we're going to look at here in just what that is here in a little bit. But if I start with one bacteria in one little place, that one bacteria is going to make another bacteria that looks exactly like him. Then both of them will make a bacteria that look exactly like them. And you end up with that little colony. Okay? When they start merging together, that's when you get genetic mutations and things like that. As we go through the chapter, we're going to look at physical and chemical requirements. A physical requirement is the way the bacteria likes its physical environment. They don't all like the same thing. That's good for us and bad for us, and we're going to talk about it kind of from both aspects. Chemical requirements are actual chemicals that the microbe needs to survive. They're going to use these chemicals to produce molecules they need to survive. So if I'm a bacteria and I'm going to replicate myself, I've got to copy my DNA. I've got to copy my proteins, my fats, all of those molecules. In order to make those molecules, I need chemicals like carbon, nitrogen, phosphorus. And we're going to look at all of those as we go through. That's kind of two different parts. This last term on here is a very important one, the word philic, or it's actually a suffix. Anytime you see a phile or philic, that is describing to you what the microbe likes. Okay, so philic is an adjective. I can't get the thing to come out. There we go. Okay. So philic, like that, is an adjective. When it's used as a noun, it's that word, phile. Okay. So to go ahead and show you how we use that word, go to our first example of physical requirements that bacteria have. And we see things like psychrophiles, mesophiles, and thermophiles. Okay? So if we just pick one to start with, the first term right here, you see how it's got the phile at the end of it? That's telling you what this microbe likes. Okay? Psychro means low temperature. Now, coming off of what I said a few minutes ago, complaining about being cold, the way I could always remember that was they like cold temperatures. That's crazy. That's psychro. So that was the way I always remembered it. The good thing about bacteria that like to grow in cold temperatures is they can't make us sick, right? Why can't they make us sick? Because our body temperature is 37 degrees. A psychrophile is a bacteria that can only grow between 0 and about 20 degrees Celsius. When you get up in 37 degrees Celsius, it can't live inside of you, so it can't make you sick. So what's the bad side? What's the downside of a bacteria that can grow in a very cold temperature? Where's it going to grow? In our refrigerator. So why do we put food in our refrigerator? Make it last longer, right? Keep it good so it doesn't spoil. Well, when you put it in that refrigerator, you're just kidding yourself when you tell yourself there's nothing that can grow in it to make it spoil. There's lots of things. Can you guys think of something you know right off the top of your head grows very well in your refrigerator? Mold, right? We've all had that big block of cheese that we put in there, and then we hadn't eaten on it for a while, and we all decided, well, I think I would like some cheese. Pull it out, and what does it look like? Furry and green. That fungus is a psychrophile. Fungi love to grow in the refrigerator. There are some bacteria that can. There are even some hyperpsychrophiles that can grow in your freezer. Okay? So a lot of people will tell you that freezer burn, you know how your meat will turn brownish gray colored in the freezer? A lot of people will tell you that's just moisture getting in your food. No, that's bacteria growing in your freezer as well. It does have some bacteria growing in it when it's freezer burn. But now, can we eat the food that has stuff growing in it in our refrigerator? You can, because once you get that in your body, it can't grow in your body. Why do we throw it away? Well, it looks nasty. If it's like a big green mold, we don't want to eat it. The other reason we throw it away is because a lot of times they start growing under fermentation conditions in the refrigerator. So they produce alcohols and vinegars, and they taste sour. The reason they taste sour is because they're doing fermentation. When you buy your milk at the grocery store, is it sterile? Does it have bacteria in it? Yeah. Why do you think that expiration date's on there? Because that's how long they think it's going to take before the bacteria grow enough that it's going to spoil your milk. Is that expiration date set in stone and marked? That's the day you throw it away. No, my husband thinks it is. If it says September 9th on it, September 9th that morning, he will freak out. We can't even drink that milk. It's September 9th. Well, I have a smeller. Are any of you smellers? It doesn't matter when the date is. Every time I open the milk, I smell it. That comes from my grandma, though, but she believes that if it smells bad, then you need to pour it in a glass. It may just be the jug that smells bad. That's the older. If you have an older person, they'll tell you the jug will stink before the milk does. I don't know. If the jug stinks, I'm tossing it. All right, so that's the psychrophile. The second group is mesophile. Meso means middle. They like the middle temperature range. They grow 25 to 40 degrees Celsius. That's room temperature and body temperature. The bad side of these, they are the ones that make us sick. The group of bacteria that we call pathogens, they're all mesophiles. Can they grow in the refrigerator? Nope. If we heat them up, will we kill them? Yes. That's the good thing. But if we get them in our body, they can grow very, very well and make us very, very sick. Okay? The last group, thermophiles. Thermo means heat. They love heat. They can grow 50 to 60 degrees Celsius. So if you microwave something, they just keep growing depending on how long you microwave it. Okay? There's nothing really that bad about those because what's boiling temperature? What's temperature of boiling water in Celsius? 100. So if you boil your water and you have some thermophiles in there, you'll kill them. The ones you will not kill are the hyperthermophiles. There are bacteria that can live in boiling water for hours. There are bacteria that can live, and protozoa that can live in lava, boiling lava inside of a volcano. Okay? So, take-home message, they can live almost anywhere. It doesn't matter how cold, doesn't matter how hot, but they can't all live there. They have a specific temperature they prefer. Okay? If we look at a graph, so this color right here, this bacteria is growing from 40 to 70. So which group is this? What's 40 to 70? That's the thermophiles, right? Okay, they grow starting at 40 all the way up to 70. So this is what we call a range. The peak of their growth is somewhere around 64, 65. What does that mean? That's their optimum temperature. That's where they grow best. So all of our bacteria we're going to talk about, they're going to have a place where they really like to grow. That's their optimum. But they don't only grow right there. They have a range. Okay, so if we look, next line right here is kind of the yellowy color. That is your mesophiles. Their optimum growth is up here around 37. It's body temperature. That means they grow best in our body. But if we cool down to almost 10 degrees, does it kill them? They can still grow. They just grow really slow. What temperature do you think your refrigerator is? Anybody have any idea? Most people don't. Most commercial refrigerators are between 10 to 15 degrees Celsius. So if I am sick and I cough on some food and then stick it in the refrigerator, eat it later, and then you come and eat it, can it make you sick? Yeah. Refrigerator's not going to kill it because it's in its range. Okay? So you're all giving me this look like, oh my God, you're making my life horrible. Well, just wait, it gets better. All right, so that's what temperature do they like? What's the answer? Depends on the bacteria. They all like different temperatures. Okay? So now let's think about pH. pH is the measure of acidity. Okay? Let's make sure we all know that what the range is. pH 7, that's neutral. That's the middle of the scale, right? As we go down the scale towards 0, are we getting more acidic or more basic? Acidic, okay? And then as we go up, top of the scale is 14. That's becoming more basic, okay? Just to make sure we're all on the same page there. 99% of all microbes on Earth, especially bacteria, like neutral pH. They can vary a little, 6.5 to 7.5. So they can live in your blood. They can live in your lungs. Because They can live in your urine. Your pH in your body varies between 6.5 and 7.5. There's none that we would classify as preferring to live in a base. Bases are really tough on living organisms. But there are a couple that prefer to live in very acidic pHs. We call those acidophiles. Some of them are bacteria. Most of them are fungi. They can withstand pretty acidic conditions. Okay? So I wrote a question on here. Can you think of anything that is type of food that an acidophile may spoil? Let me ask it a little different way. Is there anything you eat that's preserved in an acid? Pickles, fruit. Okay. Have you ever drank pickle juice? Some of you probably like pickle juice, right? Here's your homework. If you have a jar of pickles at home, pickles are preserved in vinegar. I want you to go home, take your pickle jar out of your refrigerator, hold it kind of above you in the light, and swirl it. What happens in lab when we swirl those test tubes? The little twisty thing comes off the bottom, right? It looks like the snot in the test tube. I guarantee you, I bet you money, if you go home and swirl your pickle jar, you're going to see the little bacteria swish up in the bottom of the pickle jar. Sounds gross, right? But is it going to hurt you? Does your body have the pH of vinegar? No, your pH in your body goes up. So those bacteria living in that pickle jar die as soon as they get past your stomach. They love your stomach because it's very acidic. But once it gets into your intestine, you destroy it. Okay? Just thought you'd like that. All right. And another reason it's going to be in your pickle jar is because how many of you go to the store and really want a pickle? So you buy a jar of pickles, and then you may not eat those pickles again for like 6 months before you decide you want to pickle again. Pickles aren't usually something we eat every day. So that pickle jar usually stays in there quite a while. Okay? It's in your pickle relish, too. You just can't swirl your relish. You're just eating that. All right. Next thing, osmotic pressure. This is the amount of salt you have. Now, you guys have to remember something from Chapter 4 to understand this. If I have a bacteria, this is on your last test, that is 1% salt on the inside, and I make it 5% salt around him, meaning I dump salt on him, what type of condition is this, hypo or hypertonic? Hypertonic. What's going to happen to my microbe? It's going to plasma. It's going to shrivel up, shrink, and die, right? The water's going to come out of him. So most bacteria, if you dump salt on them, what's going to happen to them? They're going to die. We preserve a lot of food by dumping salt on our food. Almost anything that is canned is full of sodium. The reason for that, it kills the bacteria that may be in there. Okay? And I think I've said this to you guys before. Before we had refrigerators, we salted meat because that kept the bacteria from growing on it. But can something spoil that has salt on it? Absolutely. That means there has to be a bacteria that likes salt. And unfortunately there is. They're called halophiles. Right? Now, I would never test you on it, but just for those that are curious, why do we call them halophiles? Halogens are one column of the periodic table, chloride, iodide, bromide. All of your salts have a chloride or a bromide or something like that attached to it. That's why they're called halophiles. Mm-hmm. Okay, yeah. That's actually a protozoan. It's a disease called leptospirosis that we're going to talk about when we get to protozoan diseases, disease of the urinary tract. It's carried in rat and dog urine. People get it because, so if any of you have ever noticed, if you've been at Walmart when they were stocking shelves, the pallets of cans are covered in plastic, right? They used to not be. Now we know if rats pee on the top of a can and then you use your little can opener, what does the top of the can do? Sink down in the food, right? You can get a disease called leptospirosis that way. But we're going to talk more about it later. That's not bacterial, but those leptospirosis is a protozoan that is halophilic. It can grow. All right? All right. Now, so there's two types of halophiles. There are obligate halophiles and facultative. I'll give you the easy way to remember this. If you are obligated to do something, what does that mean? Required. You absolutely have to do it no matter what. An obligate halophile absolutely has to have salt to survive. Those aren't the ones that usually hurt us too much. Because if we get an obligate halophile on our hands or on a piece of equipment, it can't live there. So it's very unlikely that that's going to get on our food we're preserving with salt. The ones that are most likely to hurt us are the facultative halophiles. They can live either way. So they could be living on a piece of fish or a piece of meat that we're going to preserve with salt, growing perfectly fine without salt. And then we dump salt on it trying to preserve the meat. The facultative halophile is going to just say, oh, okay, salt, sounds good to me, and just keep growing regardless of how much salt you dump on it. Okay? You guys have probably all opened a can of food that had a facultative halophile growing in it and you just didn't realize it. Next time you open a can of something, it can be anything, vegetables mainly, but soups, anything like that, if you see a film on the top of it, that's probably bacteria growing on it. It doesn't happen every time, but it probably is. If you want a little science experiment to do, go home and open a can of something and just leave it sitting out on your counter. Within 2 days, there will be something furry growing on it because a fungus really likes salt. They're facultative halophiles. So the fungi just blowing through the air, the molds, will land on that and just have a party and start growing really, really fast. Say it louder, I didn't understand what she says. Well, no, some of it's fat. Now, you may see a fatty layer on the top. If you pick it up with your fingers and smush it and it's greasy, that's the fat. If it's just slimy, it's bacteria. Okay? But it's okay because what are we going to do to it before we eat it? You're going to heat it. There's nothing grosser to me than opening a can of food and just eating it, and I've seen people do it. I've seen people downstairs in Jody's open a can of something and just eat it. No, you have to heat it. Okay. All right, so that's all of our physical requirements going to worry about. Temperature, pH, and salt. With all of them, they all require different things. Okay? Chemical requirements, all microbes have to have most of these chemicals. The only exception to that is oxygen. Okay? With carbon, nitrogen, phosphorus, sulfur, you have to have those molecules to make any macromolecule, like DNA, proteins, fats, RNA, any of those molecules. You have to have carbon, nitrogen, phosphorus to make. Sulfur is only for proteins. Okay? I used to always start this lecture with asking, why do you need carbon? What is it for? And I got a million blank stares. You need to watch more Sci-Fi Channel. Okay? The way you know it comes from Earth is it's based off of carbon. You are a carbon-based life form. Everything that is alive on Earth, every molecule it makes has carbon in it. Okay? So if I'm a microbe, if I'm going to make stuff, I've got to have carbon to make it. Okay? So that's just a given. You all need to know that. Okay? Now, so I started this by saying oxygen was different. Not all organisms have to have oxygen, right? Can they produce energy without oxygen? Chapter 5, how do you live without oxygen? Fermentation. Okay, so the oxygen's a little different. We have what we call aerobes and anaerobes. An aerobe requires oxygen, and anaerobe does not. But there's different types of aerobes and anaerobes. There's that obligate word again. What did obligate mean? Required. So if I'm an obligate aerobe, what does that mean? Absolutely have to have oxygen to live. You are an obligate aerobe. If the oxygen goes away, what happens to you? You die. That's an obligate aerobe. An obligate anaerobe, what would that be? Has to live without oxygen. There are some bacteria, if you put them in an oxygen environment, they die. Those are really unlikely to make us sick because they're not going to be alive where we're alive. Okay? The other types of anaerobes are the ones that are more likely to make us sick. There are facultative anaerobes and aerotolerant. So do y'all remember what facultative was? What does that mean? Either or. A facultative anaerobe will either use the oxygen or not. So if you put a facultative anaerobe in this room, there's oxygen in here, he's going to grow, use the oxygen. If you take the oxygen away from him, you put him in a candle or something like that where there's no oxygen in the environment, well, he says, oh, okay, you took my oxygen away, fine. I'm going to start doing fermentation. I'm just going to keep living. Okay? Aerotolerant is a little different. So I'm going to give you a way to remember that. What does it mean if you tolerate somebody? You don't really like them, but you deal with it, right? We all know. We've all had those people. How many of you are married? What is your mother-in-law to you? Yeah, so the ones that are married understand. You tolerate your mother-in-law. I better watch my mouth. You tolerate your mother-in-law. All right, so an aerotolerant anaerobe. Does he use oxygen? No. Does he really like oxygen? Not really, but he tolerates oxygen. So he's not going to use it no matter if it's there or not, but he's not going to die if you start putting oxygen on him. Okay? Now, so I really like this little picture, and I use it a lot of times to describe things. So this is our test tube. There's bacteria growing in it. They're the little black circles. The yellow is the media, right, that's the bacteria growing in. This is a broth. What does that mean? It's a liquid media. Okay? So our obligate aerobes over here are all clustered at the top of the media. Why are they up there? Because the only place you're going to have oxygen is in the air at the top, right? They have to have air to survive, so they're hanging out at the top. Go ahead and go over here. All these bacteria are way at the bottom of the tube. Which ones are they? The obligate anaerobes. They're staying as far away from the oxygen as they can because oxygen will actually kill them. Okay? Now, here's the facultative anaerobe in tube B, aerotolerant in tube D. What's the difference between those two tubes? In B, you've got more of them at the top, right? What are all of these guys at the top doing? They're the ones using the oxygen. The ones down here at the bottom, they're kind of the, well, it's really crowded up there where the oxygen is, so I'm just going to go down here and live without oxygen, because they can do it either way. The aerotolerant are spread throughout. There's none of them using the oxygen. They're just kind of hanging out throughout. They're doing it their own way regardless of whether oxygen's there or not. This last little group's a really unimportant group as far as what we're going to be discussing in this class. They're called microaerophiles. They use oxygen, but they don't need as much as any other organism does. So they can live anywhere that has oxygen, they just don't use it as fast. So we're not really going to talk much about those, but since I was going to use the figure, I wanted to tell you what they were. These are the four. You need to have a very clear understanding of the difference between them. Does everybody understand? Okay. All right. So now we can look at something called binary fission. Now we're going to get into the only math we're going to do in this class. And when I say that, some of you say, oh my God, math. This is like 4th grade math. And that's when I was in 4th grade. That was a long time ago. All right? This is probably 1st grade math now. It's math that you can all do, and we're not even going to use a calculator. Okay? Very easy math. Binary fission tells us that if we start with one bacterium, one microbe, that one is going to divide and become 2. That's easy math, right? 1 becomes 2. Now, what is binary fission? It is 1,000 times simpler than mitosis. Okay? And y'all thank God, right? There's no phases or anything like that. With binary fission, step 1, the cell gets a little longer and it copies its DNA. Obviously, it has to copy its DNA, right? Because DNA is where we store all of our genetic information. So that bacteria just kind of stretches out a little bit, copies his DNA. Once he has done that, then he just simply needs to start building a cell membrane and then eventually a cell wall around each portion of his DNA. Once he has done that and the cell wall has been completed, he splits into 2. It's much easier, right? Copy DNA, expand cell membrane and cell wall, make 2 cells. A little bit of terminology. This first cell is the parent cell. Okay. The 2 cells that come from the parent are the daughter cells. I don't know why they're all called daughter instead of son, but they're just all called daughter cells. They're the offspring. Both of the daughter cells are identical to the parent cell. Okay? The parent cell represents generation 0. That's the very first. All right? Once that one cell divides into 2, this is the first generation. Okay? Does that make sense to everybody? We're always going to start with 1. That's generation 0. That's where we start. When that one divides, that becomes our first generation. Let's see if you understand. If we go to second generation, how many cells are we going to have? 4. Why will you have 4 cells in the second generation? Because each one of these is going to split, right? So this first one will become 2. And the second one will become 2. So now I have 4. So how many cells am I going to have in the third generation? 8. How many? 8. I got good math, right? 8. How many cells am I going to have in the next generation after 8? 16, then what? 32. Then 64, then how many? 128, then what? A lot. This is when I start losing people. 256, then what? 512, very good. All right, and then 1,024. So what? These numbers are getting big fast, right? There's a very easy way to represent this growth without having our numbers to get so big. Because a lot of these microbes can go from one generation to the next in as little as 10 minutes. So that means in an hour you have to multiply all those numbers times 6, and those numbers start getting big. So we're going to use what we know about math to just display this a little differently. All right, so I have these little tables right here. So the first part of the table says number of cells. If we have 1 cell, what generation are we at? 0, right? Does everybody understand that? Don't say you don't just because we switched pictures, right? Here was our first cell, 1 cell, generation 0. Okay? Then when we have 2 cells, what generation are we? First. 4 cells with second. Right? Now the easier way to describe this without keeping up with all of those numbers is to remember that every time 1 cell divides, how many cells do you end up with? 2 cells. That's a special type of growth that I know you've learned about in some math class at some time. That's called exponential growth, based off a power of 2. So watch how neat this is. I can always use this base number of 2, and the little exponent I use above my 2 is my number of generations, and that is always going to tell me how many cells I have. 2 to the power of 0 is 1. So 2 to the 0 generation tells me I have 1 cell. 2 to the first power, 2 with 1 generation, tells me I have 2 cells. 2 to the second power is 4. Okay, now here's where I lose some of you because your math backgrounds are different. Why is 2 to the third power 8? Why is it not 6? Because 2 to the third power means 2 times 2 times 2. That's what those powers mean. But in all honesty, you don't really need to be able to do that kind of math. I'm going to let you leave it like this. You're not going to have to multiply it out, but you need to understand what this means. So where does the 2 come from? Why is the base power always 2? Because 1 cell always divides to make 2 cells. What does the exponent mean? Generation number. Okay? And we always start with generation 0 being 1 cell. Does that make sense? Okay? So what is the generation time? What do you think the definition of that would be? It's a time, so it's how long it takes to do what? Reproduce, to go from one generation to the next. Okay? Is this useful in real life? Maybe not to you. I heard a lot of people say no. Maybe not to you. No, you're not going to clean your toilet and say, okay, there's 1 bacteria left. And if his generation time is 30 minutes, that means in 2 hours, I'm going to have 2 to the 4. You're not going to do that. But in a science lab, we do those calculations all the time. So we're going to do some basic calculations in here just to get an idea of how this works. Okay? It's a very simple math problem. We're going to do 2 examples. Or actually, we're going to do this first one together, and I'm going to show you how to do it. And then I'm going to send you home and see if you can do the second one by yourself. And then next time, we'll do the second one together. All right. So let's read the problem together. And I want to start out by saying I know I'm not a math teacher, so I'm doing the best I can with teaching math. Okay? All right. If 23 S. salivarius were placed in a culture tube and allowed to incubate for 4 hours, how many bacteria would be present in 30 minutes? The generation time for S. salivarius is 30 minutes. Okay, so let's figure out what it says. What do we know from this problem? We know that we started with 23 bacteria. Okay, that's how many we started with. We stuck those in a test tube and we let them grow for 4 hours. Okay, we know the generation time is 30 minutes. All right, and we're asking how many are going to be present in 30 minutes. So what do we start with? These are very simple. We're going to break it into 2 parts. Part 1 of the problem is you need to figure out, gosh, this pen's hard to write with, how many generations do I have time for my bacteria to go through in the amount of time we're looking at? Most of you could probably just say it to me, but I'm going to show you how to figure it out. Okay? Can anybody tell me? Okay, maybe y'all can't, so let's just do it together. So how many generations do we have? So I'm going to use what information do I have in the problem? The generation time is 30 minutes. So what does that tell me? That tells me that I have 1 generation go through every 30 minutes. So everybody understands that, right? That means in 30 minutes, every 1 bacteria is going to do what? Split and become 2. Okay? Now, read the problem again to yourselves. We took 23 of them, we stuck them in there, and we just let them grow. The question is, how many bacteria would be present in 30 minutes? I'll give you a hint. One of those numbers is irrelevant. Which number doesn't matter? The 4 hours. Does it really matter how long I let them grow? I could say I let them grow for 15 years. What did the question ask you? How many are there in 30 minutes? Okay, so number 1 rule of math is what? They always give you a number you don't need just to make sure you know you don't need it. So I don't like math, to be honest. Okay, so the question asks you, how many bacteria do you have present in 30 minutes? And I'm trying to figure out how many generations am I going through. So let's think about our math. I need to multiply this number times something that has a minute in it, right? What are my minutes going to do? Cancel out, and what am I going to end up with? Generations. Does everybody understand that? If not, you need to come sit down with me and we need to have a math lesson. And I'm not making fun of anybody. I'm just saying if you don't understand the math, you need to come tell me. Okay? So how long was I going to let it grow and try to figure out how many I had? 30 minutes. So what's the answer to that? How many generations did I let it go through? 1. 1 times 30 is 30. 30 divided by 30 is 1. You really shouldn't have to do that when you read this problem because we only let it grow 1 generation. But sometimes the numbers are not going to equal 1. Okay? So I have 1 generation. To answer the question, I know that I am going to have a base number of 2 in there somewhere, right? Why am I going to have a base number of 2? 1 always becomes 2. So if this is my base number, where does my number of generations go? Exponent. So 2 to the first power is going to be in my answer somewhere, right? Does everybody understand that? Okay. Is my answer 2 to the first power? No. Why not? Because I didn't start with just 1 bacteria, right? How many did I start with? 23. So my answer is 23 times 2 to the first power. Yeah, you could calculate that out if you want to. It'd be 23 times 2, which is 46. But you don't have to. As long as you can get to there, that's good enough. Because it's not always going to be a 1 up there. It's going to get harder. Some of you look really scared. These are not hard problems. I think the reason y'all don't like them is because it's math. Oops, I didn't mean to change it. So you automatically just think that it's hard. It's not. Okay? So I'm going to skip this second one. So I want you guys to see if you can do it. We're going to do it together. And there's several examples at the end of your chapter for you to practice with that you have the answer and the question. So I want to see if you can do the second one. And this is what we'll do at first in class next week. See how many of you got it right. Okay? Do it this way. Figure out how many generations you can go through. Okay? This one says you've cleaned the toilet. You've removed all the bacteria except you got 4 E. coli left. The generation time for the E. coli is 20 minutes. That means every 20 minutes, every 1 E. coli becomes 2 E. coli. Everybody understands that, right? And the question is, how many are you going to have there after 2 hours? Okay? So you're going to figure out your generations that you go through. And so, see, some of you have already figured it out. And then see if you can answer the question for me. Okay? Now, the last thing we're going to look at in this chapter is phases of microbial growth. Now, this is kind of approaching it a little differently. What we were just looking at was how many bacteria were reacting. 1, right? We said that 1 bacteria splits and becomes 2. We can follow what's happening in that one little group, the one little colony. When we look at phases of microbial growth, we're thinking about what an entire population of bacteria are doing as we watch the lifespan of that population. Okay? So how can I make this more obvious to you? Okay? Would it be different to observe what just I was doing throughout the day, comparing that to what this entire group of people were doing throughout the day? That's 2 very different measurements, right? Obviously, I'm going to be doing something different than Louis is doing throughout the day, than Dominique is doing throughout the day, right? Okay, so when we're looking at those numbers, we were looking at what one individual bacterium was doing. Now we're going to kind of look at what a group of bacteria are doing. If we follow a group, they start from the beginning of their growth to their death, they always go through 4 distinct phases as they grow, as they divide and change. They go through a lag, a log, a stationary, and a death phase. Okay? This is a picture from your textbook. I know it's describing it to you. The best way I can describe it to you, some of you are going to think it's kind of silly, but as long as it makes you remember. So we're going to pretend we are the group of bacteria, okay? And someone has just put us in this room, given us some food on the table, locked the door, and said, survival of the fittest, go. That sounds horrific, right? We do that every time we put bacteria in a test tube. We take a group of bacteria, we put them in a test tube in some media, which is food. We seal it off and say, go for it. Okay? If we were all put in this classroom, locked in here indefinitely for the rest of our lives with a certain amount of food, would we all just say, all right, sounds great, let's go, and just start eating and reproducing and just going at it? We'd probably want to get to know each other a little bit first, right? All of us are not just going to say, well, heck, I'm going for it and just go up there and start eating. There may be a couple, right? There's always a couple people that aren't shy. Most people have to have a few minutes to get used to each other. Bacteria have to get used to each other. So when you first put a population of bacteria in a medium, they go through their lag. They're not dead. They're just not growing a whole lot at that moment. They're getting used to their environment, and they're just kind of hanging out. So when we look at a graph, I'm sorry, it's hidden behind this block, but it's a straight line. This graph is showing us as you go up the y-axis, you're increasing how many bacteria you have. So they're a flat line. Doesn't mean they're dead. It means they're not growing. They're in the lag. They're getting used to their environment. Okay? After they get used to their environment, we see this huge surge in growth. So think about what happens in our hypothetical situation where we're all locked in this room. After a while, we realize nobody's coming to get us. We've got to start eating and reproducing and growing. We've got to survive, right? So the bacteria just start surviving. They start eating. They start reproducing. This is asexual, so they're not doing anything dirty. They just start growing. Why do they not just go from a log phase, a growth phase, directly to a death phase? Notice there's a stationary phase in the middle, right? It's a flat line. Does that mean they just all grow, all stop growing? Probably not, right? What happens in real life? Think about our little room, right? As food starts depleting, do we all just say, oh, there's not enough left for everybody, so we'll all just sit here and die together? Come on, y'all play along. Doing the best I can to make this real life. What's going to happen if we have food in here? They're going to fight for it, right? Who's going to survive? The bigger and stronger people, right? And the little skinny people, they're the first ones to go, right? In our little bacteria population, the smaller, the more unhealthy bacteria, they start dying off. The healthier ones keep growing. So you'll have some of them will keep going up. I lost my pen. Some of them, if you followed the line of just a couple of them, they would keep going up. But some of them are going to start dying, right? They're going to start going down. If you've got a line going up and a line going down, what's the average of it? Flat line. So in the stationary phase, some of them are growing, some of them are dying. Eventually, no matter how strong you are, if we look at our group in here, if the food is gone, we've all been pooping over in the corner, because when you eat, what happens? You got to poop. That's just part of life, right? Okay. So we've got waste building up all in our room, and we've run out of food. Eventually, it doesn't matter how strong you are, what's going to happen to everybody in the room? We're going to start dying. And that's what happens to the bacteria. They enter their death phase, and they start dying. Okay. So now every time you get a test tube of bacteria in lab, think about those poor bacteria and what we've done to them. We've stuck them in there, and we've just stuck them in an incubator and said, here, get used to each other, start growing. Eventually, some of them start dying. Have you noticed when you look under the microscope after you stain, how I keep saying, oh, you have a lot of background? Or, oh, those little pieces of purple and pink, that's not anything, right? I've said that to a lot of you. Guess what that is? That's dead, broken down cellular components where some of them have already died. And it's just kind of pieces of the old cells laying around. Okay? So you've seen a lot of this. You just didn't realize what you were looking at. Now, here's what we're going to do with this. Do you think every bacteria on Earth will give you this exact same line, regardless of what you do to it? Of course not, right? What do we always teach in science class? This is the norm, right? This is the normal one, the average one. So let's go through our first little scenario together, and then we'll see if you guys kind of can pick up. Okay? So in our first situation, we have a halophilic bacteria. What do I already know about this bacteria? Like salt. So the more salt you dump on it, the better it's going to grow, right? Okay. So I have a halophilic bacteria. He was growing in a 2% salt media. He was growing, that's where he was. I took a few of them out, and I put him in a new media that's now 3% salt. So those of you that are my visual learners, here's my first little test tube I had. Okay. I took some of the bacteria out of that one. That was 2% salt. I put them in brand new media, except I made it 3% salt. And now I'm going to watch them grow. So what did I do to that bacteria? Did I make his life better, or did I make his life worse? I made his life better. He's happier, right? He may grow a little faster. Okay. So let me change colors since my other graph is red. See if I can make this neat enough for you to see. So what's his graph going to look like? Is it going to be identical to that one I have there? Probably not, because that's the average, right? So how is his graph going to look different? Let's go phase by phase. Phase 1 is lag, right? What's his lag phase going to look like? Shorter or longer? And why? So they still have to get used to it, so there has to be a lag phase. But it can change. It can be shorter or longer. What do you think? Shorter because it likes the salt. That's the perfect answer. That's what I would want you to write on a test. I made his life good. He said, heck yeah, this is where I want to be. So his graph is going to have a little bit shorter, that looks horrible, but a shorter lag phase. He's got a short lag phase. Now what's going on in the log phase, second phase? Okay, super fast. What's he doing in the log phase? He's growing, right? So why are y'all telling me he's going to grow faster? He's happier, right? His environment's better. So growing faster, how's that going to look on this graph? As I go from 0 to 5 to 10, I'm increasing in time, right? So it's going to be sharper, steeper, right? So he's going to really like his environment. We're going to go this little short, and he's going to grow really, really fast. All right. Now what's going to happen? He said, woof, like this. Started growing really, really fast. What did he use a lot of as he was growing really fast? He used all the salt. He used all the food. What else was he doing as he was eating and growing? Waste products, right? So his food source is going away. He's building up waste. Can he just keep growing all the way until he gets over here to the 5-hour point? He's going to use all his food faster, right? So as we go from short lag phase, really, really steep log phase, he's going to enter a stationary phase, and he's going to die. Okay? Stationary phases are probably going to be about the same length. Your stationary and your death phases don't really change a whole lot. They may start sooner or start later. Okay? Now, I will tell you that some people tell me that this is too advanced for a sophomore level microbiology class. I don't think it is. I think you guys are smart enough to understand this, right? Don't you all agree that you're smart enough to understand this? I don't like it when they tell me my students aren't smart enough to understand something. So I want you to see if you can figure out what's going to happen in the second one and draw that one. Just to talk about it a little bit before I let you go and see if you can figure it out. In the second one, you take that same bacteria and you take some from your first test tube that was 2% salt and you put him in 0.5% salt. So what did you do to his life? You made it worse. So how's his graph going to look different? Take him a while to get started, right? He's not real happy. Is he going to grow really, really fast? No, he's going to grow a little slower. Eventually go stationary, eventually die. Correct. Stationary and death usually don't change very much as far as how long it takes or steepness or anything like that. I don't think this is that complicated because I think there's only one of two things you can do. You make his life better or you make his life worse, right? If you make his life better, he grows really fast. If you make his life worse, it takes him longer to get going and grow as fast. Important things to remember. Lag and log, those are the two phases you really got to think about. And you always have to have all four phases. You can't just leave one out. Okay? Just like with human beings. We're all babies, then teenagers, then adults, then old people, right? Nobody just gets to skip teenage years. Right? We all have to have each phase. Okay? Unfortunately. So here's what I want you guys to try to do for me.